Okay, hello, we're gonna do chapter 15 and 16, gene expression and control. Let's begin. This is a review. RNA is made of ribose. DNA has thymine. RNA has uracil. These two are what's different. RNA is one-stranded. DNA is double-stranded. DNA is made of deoxyribose. So, central dogma. So, genes code and RNA are protein products. So,
DNA dogma is just DNA. It's DNA to RNA to protein.
Yep, this is what it is. And the two processes is transcription and translation. So we have mRNA, which is what sends the message from the DNA to the protein, and you have things like gene expression, transcription, and translation. Now we're going to deal with proteins and how we make proteins with DNA. So gene expression, which one does not belong? Which one? I think that's just a title, but we have DNA, then it turns into DNA. We replicate that all the time. Transcription, RNA will copy what's there, the message it's saying, and then we'll translate it into a protein. Provide one description of transcription. So let's start with transcription, which is from DNA to RNA. So DNA to RNA. So what we're going to do is we're going to make a complementary base. What that means is GC, CG, because we know guanine and cytosine pair together and thymine for DNA, but you see here uracil and adenine bind together in RNA. So here it would be GCUA. That would be what the complementary pair of RNA would be. Transcription begins at the promoter. So we have a promoter RNA polymerase. This RNA polymerase will open up a section of DNA and it'll add nucleotides to an end of an RNA. So it'll open up and then RNA will form. Transcription initiation. So RNA polymer. So we have a promoter which opens it. DNA polymers will come in, and it won't fill both sides. It'll only do one of the bases. You see this bottom base? It'll just fill it up with the complementary, which will be almost the exact same as the top, but instead of thymine, it'll be uracil. So there we have initiation, elongation, and termination. All three. This is also for transcription and translation. Both of these go initiation, elongation, and termination. So let's start with initiation. We're gonna start with the promoter. We'll break the double helix, and then RNA polymers will start. Elongation is that the DNA polymers will start making the mRNA using the DNA as a template, and it's complementary. So, like again, if the DNA sequence is ATCG, it'll be UACG. And then we have termination. We close back the double helix, and the RNA polymers move somewhere else. So, post-transcriptional modifications. This is, there's important stuff and there's useless stuff. All these codes right here don't all code for proteins. The important stuff, which stays, is called an exon, and the useless stuff are called introns. So, introns are removed and exons are used. You can have also polytails, which are adenines added to the end of mRNA, allowing for its export from its nucleus. So I think this helps it move, maybe. And then this is like a cap structure. So let's talk about genetic code. So when we remove all the introns, we're left with the exons, and these exons may carry information to make proteins. This is what differentiates between exons and introns. Exons make proteins, introns don't. So genetic code is made up of 64 codons. So each gene is made out of 64, and each codon is made out of 3 nucleotides. Each codon makes a amino acid or a stop codon. So let's use this chart. So AUG, we're going to start with A, U, G. And we get methionine, which is the start codon. Let's try UAA. So let's find U, A, and then there's another A right here. UAA, stop codon. AAG, A, G, which is lysine. And what would be an example of a silent mutation? Giselle?
Like if, well, let me see. Like if one of the stop codons changed. Like if you put a stop codon where a stop codon is not supposed to be.
No, I mean, I'm talking about like a silent mutation. Silent.
Yeah, I forgot what that was.
Okay, so it would be like if the G for a lysine, you know how it's AAG? If that G turned into an A, it'd still make the same thing, so it has no effect. Because AAA and AAG make the same thing, so if a mutation happens and the G becomes an A, nothing would happen. Because lysine will still be lysine. Glycine would be GA, glycine. Again, if the third codon changes for any reason, then it would be a silent. And then serine, or UCA, would be serine. So we...
Did we talk about silent mutations in the last?
Yeah.
Cool.
Yeah, I think it was either the last one or the one before that. No, actually, it was the last one. Or it was just yesterday. Okay, we have translation, which is now using the mRNA to make proteins. So we have ribosomes. We have... Hello, Noah.
Yo, what am I miss? What chapter is this?
Chapters 15 and 16. I posted chapter 14, which was the DNA replication stuff.
Mm-hmm.
So you said that was like the hardest stuff. So if you want to watch that video,
It was. I went over like videos and heard lectures. It was kind of easy. Like, do you know most of it?
Yeah.
Like, if I were to say right now, would you tell me if I'm wrong?
I think it was like most of the pictures were unnecessary. Like half the pictures were unnecessary.
Because you get a DNA strand, you use a helicase, an enzyme to split it open, and then it creates a 5 to 3 strand, a 3 to 5 strand, and I think the 3 to 5 is the leading strand, right?
It's about positioning. The leading strand doesn't have to wait for it to work, so it just moves continuously.
I know, but like...
Yeah, it's 3 to 5.
It's 3 to 5?
Yeah, did she say it was 3 to 5?
I think it was.
Sure.
It moves from 5 to 3, or even.
I don't think it's 5 to 3.
Yeah, 5 to 3 doesn't seem right.
And that doesn't make any sense. Because I remember telling my friends it was like 35, like 3 to 5.
Yeah.
Like, small to large.
We'll just make a little detour and.
I don't know if you can like Google it.
No, like I'm here already.
No, you should Google it because looking at the pictures is not gonna help. Hold on.
No, I think it did say.
Yeah, like...
Here, right here,
Yeah.
5 to 3. It is a 5 to 3.
And then Primase makes primers for the DNA polymerase, right?
Yeah,
For it to
this
work?
is a 5 to 3. It moves actually 5 to 3. That's what it says here.
No, that one's a lagging strand.
Ah,
He's right. It's 5 3.
no, the leading strand goes 3 5.
DNA polymerase adds nucleotides to the end strand, the 3-edged strand in the 5 to 3 direction. Lagging
Lagging goes 5 to 3. This is the thing. Lagging goes 5 to 3. leading goes 3 to 5.
And it makes like Omiyazaki or some shit like
Omiyazaki fragments, which is like
those are the gaps, right?
Yeah, they miss a spot.
Omiyazaki fragments, they come first or whatever.
RNA primer fills it in, but the DNA polymerase misses the spots, so DNA ligase fixes it.
Yeah, ligase like fills in the gaps, right?
Yeah, because look, you see DNA polymerase 1, it's breaking the primer, so DNA ligase can fill the gaps. Let's go back
to... Not that hard.
Yeah, it's not that hard. Let's go back to chapter 15.
Are you guys worried about this test?
Yes.
I don't have any need to worry anymore.
Yeah, me neither.
I'm not even really studied. I'm not even done whatever study guide you posted.
It's the same study guide stuff.
I go over like the hard subjects that were hard for me. Everything else, I feel like once I see the question and the answers, it'll like come back to me.
That's what I taught myself, and then I got a bad grammar lesson, so I don't really feel confident in this.
Yeah, and I want that A. So
I know I need to like. That's why I'm here, but sometimes it's like.
Yeah, we want.
Like most of this I know, but it's like not super crazy, I guess.
I don't know. I need to get 90 on this final today, but I'm not gonna expect myself to get that.
Yeah, I mean, if you want the A, you try it. If you content with the B, then
nah, dude. I think you gotta get like. You have to do way better before the final to get like.
Yeah, that's why they always say like if you're gonna do a class, you always have to like start strong. Like do your best grades in the beginning. So that by the time it's final time, you have a lot of room to like fuck up.
Right now, I have no room to mess up.
Yeah, so like.
I'm not gonna say I have a lot of room to fuck up because. I can only miss, like, I missed more questions on the last exam than I can miss on the final. So that's why I'm kind of worried.
But that was a lab exam. That's not multiple choice. That too. But you did also got more wrong in the last lab exam. I mean, lecture.
For me, so...
The lecture exam too. You did get like a 72, I think?
No, I got a 76.
76, so you were like right there, almost there to B.
Yeah, I didn't even study for, or we did actually study for that last.
Yeah, we did.
Because it was a lot of just second guessing myself. Like the answer was like plain and simple.
Yeah, I'd be like, oh, it's B, but then I'd be like, I don't know, B, and then I mess up and it was B.
Yeah.
I don't know. I just feel like I want to trust myself, but then I look at it, I'm reading it again, and then I doubt, and then I'm scared if I don't double guess myself, I get it wrong, and...
But the lowest I can do is get a 60% on the grade, or the test, because that's the lowest a B can be.
Yeah, I think for me, I can get like a 60 or something, and still gonna be in this class.
Yeah, but it sucks, because I want an A, but it's hard.
Yeah, I want an A in this class too, because bio's like... Do you guys have to take bio too, any of you guys? Or?
I think so.
Which one's bio too? Like...
I'm not even registered for it.
Hey,
I'm doing nursing, and bio's not one of like the major classes I need to get an A in. It's like anatomy and chem for me.
Oh, you don't even... Oh, yeah, huh? I don't even know if you have to take bio.
I do have to take bio for the prereq, but like it doesn't have to be a good grade. Like a B I need, which is...
So, if anybody wants to take like next level bio, maybe we can do something, get the same professor in spring or something.
I think it's...
Oh, you mean like how's it collective?
I mean like
I don't even know. I kind of want to take bio 2 online, like
but it's not fun
online.
Oh, micro's easy.
Micro's easy?
Really?
I thought it would be like know with a good professor, so I really don't know what to tell people when they ask me that.
Do you remember the person's name?
No, I didn't take. I took it years ago at a university, so.
Yeah, because like I'm thinking micro, and I'm thinking about this last lab exam with all the pictures. I'm like, wow.
Oh no, micro's different. For lab exams, it's all active stuff. It's not any like paper. It's like you have to gram stain, inoculate, and then they walk around looking at you doing it.
Oh.
Yeah.
I thought you had to identify pictures.
I like lab practicums. I don't like lab exams. Practicums are more like you put it
on hands.
Yeah, it's like active exams. Like for anatomy, right? All of our practicums were like
But we
like
can't
you had
do
to
that
label
here.
the cadavers, you had to label, they had different models placed about the room, and you had to identify what was in the model and all that
We
stuff.
can't do that here because,
Do
like...
you think anatomy was harder than bio?
No.
No, right?
Bio is like, bio
It's annoying.
hard.
It's general.
Yeah.
I don't even like this class. Like,
It's
Me.
general.
I-
Like, anatomy
It's
is
not even
more specialized.
yeah.
Anatomy is more specialized, so there's less to go over. So you get to go over in more detail.
Wait, for which class?
Anatomy, it's more detailed because you're
It
only
is not,
focusing
not
on
really.
You're only focusing. Oh, I mean not detailed, specialized. I mean, because you're only focusing on the human body.
But anatomy really is just a lot of memorization. Like, that's all it is. You can really count on that when you take anatomy. Everything is just direct memorization. Physiology is where it's more like this class. So, you have to know like processes and all that kind of stuff. But physiology was like hell for me because I had a shitty professor and he was just really bad. So I had to drop this class, so I had to actually take physiology at some point. I might have to take anatomy again, but we'll see.
We'll see.
What's your major?
I finished my bachelor's. These are all just prerequisites for my master's.
Oh.
Yeah.
That's
I have
nice.
other things that I have to do. Not really, because like I kind of kicked myself for not having done all this crap like years ago. Like,
At least you're doing it now.
yeah, I just wish I'd spend my summers doing more of this kind of stuff. Because then I
Really would take this
finished
in the
already and not
summer.
have to worry about it, and I would have been just working instead.
I don't know about taking
Yeah,
this class in the summer.
Huh?
This one would be hard to take in the summer.
I mean, yeah, but it doesn't matter. You can speedrun anything you try. So.
I don't know. Like, science classes are hard to take in the summer.
I mean, like, if you're not working or anything like that, it's not that hard. Like, because all you have is time on your side. So if you just, like, what I used to do, I just go to the library and study, that's it.
I know someone that does that.
And then you just study.
Yeah, just go to the library. 7 hours.
Yeah,
that's sad.
sad.
Like, I wouldn't do that. Like, I couldn't do that. Like, me,
I mean, once you get, like, once you get older, you'll understand.
Okay.
But yeah, basically.
You think it's time to go back on topic?
Yeah.
Okay, translation. We have ribosomes is where translation happens. We have tRNA, which transfers amino acids. We have the large subunit and the small subunit. We have translation RNA to protein. So our first step is tRNA carries amino acids to the carrying the amino acid start codon to the complex. Then the tRNAs go to the anticodon base pairs with the second codon in the mRNA. So they attach themselves with the mRNA, and then these amino acids form peptide bonds. And one exits, one enters. So we're gonna go more in detail. So let's start with initiation, which is the tRNA bonds with the start codon, which starts the entire thing. Elongation, things enter, things leave, things enter, things leave, making the protein longer.
What is that? Wait, what is all these steps like?
It's the same for
translation
and transcription. It first starts with initiation, elongation, and termination. So the first one is the tRNA starts the start codon. Then it elongates by adding more amino acids, and they form peptide bonds.
Okay.
And when one comes, they leave. Once they detach their amino acid, they leave. and then termination, a stop codon starts, and then they release.
And what is translation for again? Sorry.
Translation is from RNA to protein. Transcription is DNA to RNA.
So it goes DNA, RNA, protein?
Yes.
And it goes transcription, then translation?
Yes.
Yeah, transcription and translation. Just remember that C comes before L.
We talked about the mutations. We have substitution, insertion, deletion. I'm not going to spend time over that because I think you were here and Giselle, you were here. You guys understand this. Anemia. Oh, this is an example of base pair substitution. Now she actually gives us a substitution. So in this one, one of them gets substituted, so hemoglobin turns into a sickle cell. This one is deletion or insertion, which is beta thalassemia. And it's really, you lose your entire frame shift, so a lot of things go wrong. So which one is A, B, and C? Oh, she gives us the answers already.
Yeah.
But why is A substitution?
Because... Where is it? C substitutes G?
Yeah. Why is insertion B?
Because there's an extra C.
Yeah, and what happens to the entire thing?
What do you mean by the entire thing?
Like, what happens to the entire...
Insertion?
Yeah. Remember, codons come in threes. Codons come in threes.
I don't know.
So, in substitution, it would have been CCA, ACT, and CGA. The ACT and CGA amino acids would be the same, right? And then CGC would be different. I mean, CCA. In this one, we have CCG, AAC, TCG, and we only have an A. So let's look. Our first one is CCG, which is different. That's one wrong amino acid. AAC is different from ACT. That's another wrong amino acid. And TCG is different from CGA. So all three of these amino acids are broken. And same thing for deletion. We have CCG, AAC, and CGA. How is this deletion? The T gets deleted. And this is also another example of insertion because we just added a C and removed a T.
What can these cause? Like mutation?
Mutations. These are mutates, the different types of mutations. Now let's talk about gene expression. So, this is a little confusing to me. Yeah, gene control. Then after gene control, we have this. This is not fun. So let's talk about gene control. I understand in bacteria, typical cells use 10% of its genes at a time. So we're never using 100% of our genes. The same genes, but activation. So remember stem cells? What they do will turn off their genes to become specialized. Ways to control. We have transcription factors. Protein that influences transcription by binding to DNA. So transcription is DNA to RNA, so by not allowing the mRNA to form, we can stop it.
Oh my god. Yeah, sorry.
Oh, it's okay. We have the master gene, which...
What happened?
I'm not.
No, I was doing something.
Oh, no.
It's okay. Master gene is the master of all genes.
Mm-hmm.
Knockout. So we'll just... Intention manipulation of a gene to inactivate it. So you purposely inactivate it. And homoetic genes control development by the same mechanisms across species. So these are like... In the embryo, things start producing at a rate. So this is gene expression. So let's talk about bacteria, because bacteria is nice, easy, clean. So we have DNA, RNA polymers, and it wants to get these lactose enzyme genes. If there are no lactose, there's no purpose for these enzymes. So what happens is when there's no lactose, this doesn't react with it. So what happens when we don't react with this? This becomes a wall. When this becomes a wall, this RNA polymers cannot reach these lactase enzyme genes. Let's see if it's on. If it's on, lactase will bind with it, and it'll allow the polymers to reach the lactase enzyme genes. If there's lactase, the genes work. No lactase, there's something in the way. There's gene regulation, and it doesn't work. So why do we need multiple ways to control gene expression? What does this picture? Oh, this one is okay. I think. Okay, cells control the frequency of transcription. Different mRNAs may be produced from a single gene. If an active protein is an enzyme or catalyst. I think these are all the ways it can regulate itself. I just don't know how to explain this, really. So, chromatin and chromosome organization. So, we talked about this.
Yeah, can you go up a little bit?
This picture?
Yeah, some more. Some more, please.
More? This one?
Oh, do you guys know the differences in mRNA or the RNAs? Like, tRNA is a transfer, right?
Yeah, tRNA transfers amino acids.
What does mRNA do?
It's a messenger. It sends what the DNA... because DNA can't move outside the nucleus, mRNA can. So it'll copy what the DNA wants, and it's the exact opposite, but that's how DNA works.
Yeah, it's what you get after transcription.
Yeah.
You get that cool mRNA.
And then that mRNA leaves the nucleus to the ribosomes where translation happens.
Okay, go to the next one.
Which one?
The one after that one, after controlling gene expression.
This?
No, go down.
The bacteria one? This?
Yeah, keep going.
This?
The one after that one. Yeah. Okay, yeah.
That's where we were.
No, before.
This?
Yeah, the one you said you couldn't explain.
Oh, yeah, this.
Yeah. So those are just showing the ways that...
Like, different ways to gene regulation.
Yep, yeah. Gene regulation. Okay, cool. I just wanted to see what that was about. Okay, you can keep going. Thanks.
Okay, chromatin and chromosome organization. So, we have DNA, double helix. We can form these with histones to make nucleosomes. This is weird.
No.
This picture looks weird.
It's showing you how, like, everything gets organized and made into a chromosome.
But, like, I liked, is this picture here? In this one? No, it's not. I like the other picture better.
There was another picture before, yeah.
It
was on yesterday's slide.
Yeah, I like that picture better.
Okay, keep going, yeah.
DNA methyltransferase groups of enzymes add methyl group. So methyltransferase add methyl to cytosine.
So if you have high levels of methylation, that means it's not going to be transcribable.
Okay, that's cool. So histone modification is you got adenyl group, methyl group, phosphate groups, and short peptide chains. Histone is a protein that like rounds things. So.
Yeah, so like the DNA is made like wrapped around histones and stuff.
This stuff is just confusing.
So all acetylation, it's gonna decondense chromatin, which means it's gonna release those histones in order for transcription to occur. So methylation does the exact opposite. Make those histones get like tighter and stuff like that.
Thank you. And maybe I remember her saying something about this, like when it's open like this. Yeah, if it's open, then there's a lot of proteins and stuff being made, but if it's closed, then you can tell this probably is dying. Not making the highest. So chromatin remodeling complex. So we're going to use ATP to reshape chromatin.
That's just another way to enhance the ability to transcribe.
Yeah.
So you're able to knock off those histones and the DNA is able to unwind some more so that you can further transcribe more DNA.
We're on the last 3 slides. Okay, gene pathetic inheritance.
Epigenetic.
Rides a way for each daughter cell to inherit patterns of gene expressive, selective term for any mechanism of inheritance. This is do something other than differences in DNA sequence.
So how you can understand this as an example is there are these mice, right? And they're obese because people give them like an obese diet. So once they have their little mice babies, those mice babies are actually predisposed to like risk factors and stuff because their parents were like given like a lot of like fatty foods and stuff like that.
Oh, that's cool.
So, it's like alcoholism, stuff like that. Like you're more prone to become alcoholic if your parent was alcoholic, like crap like that. So yeah.
So let's talk about bar bodies. It's when we deactivate one X chromosome, so we only have one pair of X, because males don't do this because we have an X and a Y. Girls do.
Yeah, girls are gonna have bar bodies because
two
it's
X's.
gonna be redundant for us to have 2 X chromosomes that are active. So one is always inactive. That inactive one is called the bar body.
Yeah. And then post-transcriptional control, we can use mRNA, which is mini RNA, to like close it before it like reaches everything.
Yeah. So what mini microRNA is gonna do? It's gonna attach to the mRNA, and then it's going to make it be able to be destroyed. It can't, so that it can't be subscribed upon because that microRNA is already attached.